Hello friends, we will be studying about the antibodies in this lecture, and we will see what are the antibodies, where are they produced, the structure, the types of antibodies, and finally with the function. You know that antibodies are the warriors of our body that helps the body to fight against the invading pathogens. They are the globulins, also known as the gamma globulins, which are the key component of our immune system. And these antibodies are usually produced in the B cells. You know, our body has two different types of immunity, the cell mediated immunity and the humoral immunity. In the cell mediated immunity, you will find the first line of defense like the macrophages, the dendritic cell, and different other cells. And the humoral immunity is the important component because in the humoral immunity the B cells are produced, and these B cells after maturation will form into plasma cell, and you know these plasma cell are the antibody creating factories, and these antibodies are produced in this plasma cell and they are transported with the help of bloodstream to the different parts of the body. So let's see the structure. The antibody has a Y-shaped structure, as you can see here, and they have two different chains. One is the heavy chain and the other is the light chain. And this heavy chain are of two identical heavy chain, and also the light chains also has two identical light chains. And these heavy chains and light chains are divided into two different regions. One is known as the variable region, denoted by FAB region, and the other is known as the constant region, denoted by FC. And this variable region is used for the interaction with the antigen, and the constant region is used for the involvement of cell surface receptor of our immune system. And these heavy chains and light chain has different amino acids in it, and these amino acids are attached with the help of disulfide bonds, and these chains are repeated and it varies according to different immunoglobulins. The FAB portion or the variable region is in direct contact with the antigen, right? So it changes its structure and it varies its structure according to the different antigen present. So the amino acid sequence present in this variable region also changes accordingly, because we know there are different types of pathogen and that used to interact with our body, and the body doesn't know what type of pathogen comes. To remove the limitation, the antibodies produces different types of antibodies, so the amino acid also keeps on changing due to different types of mutation, also known as somatic hypervariable mutation. The constant region, also known as the FC region, doesn't change. It remains same throughout without any mutation. Okay. The light chains are attached to the heavy chains with the help of disulfide bonds. There are different types of linkages overall, that is between heavy chain, and also linkage between heavy chain and light chain. And there are some other linkages, that is intra-disulfide linkages between amino acids. Okay, you can see here. So now coming with the types of antibodies, you see there are basically 5 different types of antibodies, and each consists of 2 identical heavy chains and 2 identical light chains also, and some of them may form dimers and pentamers also. So the first type of antibody is the immunoglobulin A, which is a secretory antibody. It has 2 different subunits and they remain attached to form a single unit, and it is linked by a joining chain in the middle, which will bind both the unit into a single unit. And this type of antibodies are usually found in the saliva and the secreting organ, or the secreting fluids like the saliva, tears, breast milk, and they act like a first line of defense against the pathogen. Therefore, it is known as the secretory antibody. The second type of antibody is also known as the immunoglobulin D. This is very similar with that of the immunoglobulin A, but here it has a single unit without any subunit or a dimer. Immunoglobulin D also acts on the mast cells, and it causes various types of interaction like the allergic reactions. The other type of antibodies, that is the immunoglobulin E, is a type of antibody which has some hypersensitivity reaction. The immunoglobulin E function is to involve in some hypersensitive reaction like production of allergens. Okay. They involve in type 1 type of hypersensitive reaction through its constant region, which can involve in the interaction. And this immunoglobulin E also can fight with the different types of worm present in our body. They used to coat the worm all over their body with the help of these immunoglobulins, and then start interacting by bringing some cytotoxic cells. They will trigger some responses in the cytotoxic cell which can create pores in the membrane walls of this worm and finally kill it. Immunoglobulin G is the another type of immunoglobulin that was present in our body from the time of our birth. And this type of antibody, that is the immunoglobulin G, can transfer through the placenta to the newborn baby. The final type of antibody, that is the immunoglobulin M, which is present as a pentameric form. These pentamer are joined with the help of joining chain and disulfide linkages between them. And this immunoglobulin M is also a common type of immunoglobulin that are found in the B cell receptors. Okay, and this immunoglobulin M is also used for fixing complement. Now coming to the functions of the immunoglobulins or the antibodies, the main function is to recognize the antigen. You know, antigens are the foreign substances that is capable of activating a immune response. These immune responses can be from viruses, bacteria, fungi, and also may include non-living substances like toxins, foreign particles, and also chemicals. These antibodies will trigger chemical responses from the antigen to the body to remove the antigens. Okay. So one such main function of the antibodies is to neutralize the antigen. That is, if there is any pathogen inside the body, the antibody will form a complex by binding with the antigen, and they will try to neutralize the antigen by masking it. There is some other function as well, like opsonization. Opsonization means whenever a whole body of the pathogen takes entry into our immune cells or into our body, the pathogen will be coated by the antibody, so that the phagocytic cells will find easy to engulf the pathogen and kill it. Signal transduction is another function of the antibody. There are some subclass of immunoglobulin G which can involve in this type of signaling, known as signal transduction. Finally, antibody can also involve in antibody dependent cell mediated toxicity. This is another function where the cytotoxic T cell can recognize the constant region of the antibody, and it will start releasing some chemical factors that can easily kill the cell by cell lysis. So this is what about the overall idea of the antibody. So I hope you understood it well. If you understood my lecture, please hit a like, do subscribe to my channel, and also share with your friends.